And so we're going to shift to another story that's about really small stuff,
this one's awesome.
which is now coming out of Yale University. So the story out of Yale is Yale scientists recode the genome for programmable synthetic proteins. A team of synthetic biologists have rewritten the genetic code of an organism using a novel cellular platform for producing a new class of synthetic proteins. Very fascinating. A little bit earlier this year, this came out. And so, help us understand the implications of the ability to recode the genome for specifically programmable synthetic proteins.
Yeah. We've talked about synthetic bio on this podcast before, like the power of CRISPR. This takes it even a step further. Okay, this is like, honestly, I thought this was kind of crazy when I read it. Okay, so CRISPR is the programmable DNA tool, right?
Yes.
That's the thing that lets us snipe a bit of DNA here, and then other enzymes will come in and put in whatever custom bit of DNA we want, right? This thing is making so with CRISPR, one can imagine that this genome sequence was maybe possible without humans, right? Through mutations and stuff like that.
Yes.
Here, we're making synthetic biology that literally could not be possible without humans.
The idea is CRISPR, you can see natural processes get the result of what we can.
Yeah, maybe in a billion. Yeah, it'll take forever, right? Here, we're so CRISPR, we're using the same Lego blocks, right? ACTG, right, that already exist. We're just reprogramming how the ACTG is said because we know what the code is, and then we can create our own proteins. Here, we're creating new Lego blocks.
Entirely.
Does that make sense?
So we're adding more letters to in this analogy, into the ATCG.
Not to the DNA, but to the proteins.
The protein creation and synthesis itself.
So let's go over some basic molecular biology, okay? Because that's what's going to be needed for this.
Okay.
So DNA. DNA comes in 4 letters, okay? There's 4 building blocks that make up DNA, and DNA is essential to all life, right? That's where we get the genetic information from, and that tells us the blueprint of how to create whatever we want to create. Okay, DNA comes in 4 letters, ACTG. These are nucleotides. They're just like tiny molecules. They've got 3 parts, and you build a ladder on top of each other, and you create a giant little, staircase of
the double helix.
Right? And then the unique DNA, you have unique DNA, I have unique DNA, and that determines the kind of organism you're going to be, the facial features, everything.
It determines whether you have good genes or not.
Oh, yeah. Yes. Those are the genes we're talking about, people.
Not on the body, in the body.
No, in the body. And so DNA is pretty standard. 4 things. Proteins, on the other hand, there's 20 different amino acids that life uses. Okay. So 20 different building blocks.
Got it.
All right. Now, what DNA does fundamentally is it tells the cell which proteins to put in a sequence. Okay. And what these guys have done is created new amino acids. Entirely.
Beyond the initial list of 20.
Yes.
Oh, wow.
That's okay. You see what I'm saying?
No. We invented a new Lego block.
Yes. We invented a new Lego block for proteins.
That's actually okay. Which is the implications of which now. Okay.
Yeah, we can let's discuss the implications. First, let's get into the science, which is because
this
experimentally, this is such a hard thing to do. Okay, and I'm going to get into why. So first, why even do this in the first place? Right. Why does it make sense to do this? So here we're going to get into a little bit of mathematics, okay? So there's four letters in DNA.
Yes.
But I want to code for 20
amino acids. Yes.
Let's say 20 distinct words.
Okay, sure.
4 letters.
4 letters, 20 distinct words.
20 distinct words. Now, I can't just have one letter per word.
Right.
Because then I'd only have 4 words.
Yep.
ACTG.
Yes.
Okay? With 2 letters per word, I would have 16, right? I can have AA, AT, AC, AG, and so on and so forth. So 4 times 4, 16 words.
Yep.
With 2 letters.
Yep.
But with 3, I have 4 times 4 times 4. That's 64.
Yes.
Yeah. So I can do 64 different words.
Word combinations with those.
But I only have 20.
Right.
So I need a minimum of 3, but that means that there's a bunch of repetition.
Right.
Right? Because I have 64 different possible words, but only 20 different meanings, so to speak, right?
Yeah.
So multiple words are going to mean the same amino acid.
Yeah.
Yeah? And so there's going to be this redundancy.
Yes.
And life codes for that, right? So for example, there's three three-letter words, TGA, TAA, and TAG. All of these three code for stop making a protein. Okay? ATG, so like you can imagine there's a little like enzyme called RNA polymerase that like goes down and it tries to find ATG, okay? That becomes AUG. I guess it tries to find AUG in mRNA. mRNA uses a different set of letters. Instead of T, it uses U. Anyways, there's a little like guy who goes down and he's trying to find AUG, okay? Once he finds AUG, he's like, oh, a protein starts here.
Got it.
Then he recruits a bunch of other stuff. There's a ribosome that comes in, which is the protein making factory. Ribosome attaches over there. And then now the ribosome goes, okay, what are these three letters? Okay, I need this thing. Okay, next. What are these three letters? Okay, I need this thing, right? And so it keeps going until it reaches something called a stop codon, which is a signal to stop.
Yes.
That's the end of my protein.
Yes.
And there's TGA, TAA, and TAG. There's three different words that mean the same thing, stop.
Right. And like in theory, I think I can see where we're going with this, because what we're sort of saying is there's this natural redundancy where certain combinations all equal the same action. We don't necessarily need all of those to have the same action because we already have the action.
Yeah.
So
we just need one.
Yep.
We just need one to reliably mean stop.
Yeah.
And then the other two, what if we coded some crazy meaning?
Yeah.
Right? That's never been seen before.
Right.
So what if I created an amino acid that's never been seen before that has unique chemical properties, and then I started using that to create proteins that have never been seen before?
Right.
Not even in the millions of years of biology.
Yes.
Right? In the trial and error.
Yes.
Because so proteins are effectively this chain of amino acids, and on the back of each amino acid is something called an R group. I think it's called a residual group, but I might be getting that wrong.
They'll get us
in
in
any
the
case.
comments.
Yeah, they'll get us in the comments. The R group is basically the thing that makes the amino acid what it is. Sometimes it's like a sulfur atom. Sometimes it's just a lame carbon atom or a hydrogen atom. Sometimes it's like a phosphate group, which has a charge.
Okay.
Right? So you can have like a bunch of things that have charge, and then those charges on either side will start folding the protein, and that's what gives it the 3D structure. All of these R groups interacting in this 3D kind of way is what folds this 1D chain into a 3D molecule of a protein that like makes a protein do what it does.
Right.
But now what if we can have different R groups that like with different elements or like completely unique properties that start making the protein do crazy things?
Because what's interesting is you have the chain, but to get the effects, it needs to be into the folded 3D structure. And sort of what we're now saying is, in the wake of something like this, as kind of like a side note, in the wake of something like the Google's product that they put out, which created the library of all of the different protein fold structures.
Yeah, Google AlphaFold. It won the Nobel Prize in chemistry last year. Right.
Yeah. And so what you're
they're actually using some AI to like try and figure out what effect this kind of stuff would have.
But even if we can see what the protein effect is, what this is sort of saying is we're actually changing the factory that builds them to do it out of the box effectively and create these new protein structures which have these emergent properties that can be completely net new than anything we've ever been able to experimentally
see. I think this is even like CRISPR gives us the ability to make new DNA.
Right.
But like new DNA in terms of like with the same letters.
With the same Lego blocks.
This is like new proteins with like, you know, like
who knows what?
Like we could start imagining now we can use a full vocabulary of 64.
Which if we started with 20, and we now have 64,
look at the diversity of life that we get with 20.
Let alone now almost tripling, right? Over tripling a number of, again, fundamental ways in which you can generate and this is actually really
it's so nuts.
Really
it's
crazy.
So nuts. And like it was actually like so hard to do.
I mean obviously.
Okay, it's like so hard to keep a bacteria alive and also like mess with it so much that like, you know, this poor bacteria is like, bro, like I have no idea what's going on. Right. So what they did was they first started with a strain that already had one of the, I think TAG was, so TAG is one of the other stop codons.
Yes.
already completely eliminated. Okay. They had just like done CRISPR enough to remove all of the TAGs. Okay.
In a separate
in a separate context. In a separate context. So they're like, let's start with that thing because one of them's already done.
Because someone removed it and everything worked fine. Is what we're sort of saying. So it's like we feel comfortable that if we recode it for something else, there are not going to be any negative derivative impacts of the removal.
Yes.
That's already been taken care of.
Yeah. So next, what they did was they said, okay, now I've got two other stop codons, TAG and TAA. What I'm gonna do is make TAA the stop codon. It's the stop codon. Okay, that's the thing that's gonna tell something to stop making a protein.
Yes.
TGA is what I want to repurpose for something else. Okay. So first thing, what you got to do is there's something called so how does the cell know to stop protein making once it sees this? There's these things called release factors that are proteins in themselves.
Yes.
And they look at, oh, there's a TGA. Okay, everybody stop what you're doing. Yeah. Okay, so they got that protein, kicked it out.
Yep.
They got the gene for that for that thing. They booted it out of the genome. So now that thing doesn't. So that like policeman that's there to like say, hey, I see a TGA, you better stop. Yeah. That thing's gone.
So we've defunded the police.
Yeah. So we defunded the TGA police.
The TGA police.
Right. And we've kept the TAA police.
Got it.
Okay.
Got it.
And we've deleted a bunch of non-essential genes, so we've made the genome a lot shorter. This one's interesting. So there's 3 genes in the E. coli genetics where TGA happens in the middle. Okay. But like something gets put in instead, because TGA is sometimes a stop codon. Because biology, there's never a hard, fast rule. It depends on the context of where this codon is in terms of the rest. So there's 3 very important genes that has TGA in the middle. So they were like careful enough not to completely remove all TGA.
It wasn't a blanket delete. Yeah. It was a delete except for
and the other parts weren't a delete. It was a substitution. Instead of TGA, they put in TAA.
Got it. Got it.
Yeah. So except for this part, because if they put a TAA, then that protein is done, and then
that makes sense.
Right. So. Even that they're targeting, like, you can't see this. You're not like reading it going, I want, like you got to make some molecular machinery and experimental apparatus to actually target the correct ones, leave out the ones that are important. Yes. So they did that. Then they ran into some problems, which is release factor 2, which is the TAA police. Yeah. TAA police, because was starting to get confused, because it started not only like it started recognizing the wrong thing. Yeah. Okay, because we've messed with this thing so much. Right. It started sometimes not recognizing TAA because we put in so much of it. Yeah. And then somebody would put in a tryptophan amino acid there instead of stopping the protein production.
Got it. Yep.
So then now they're like, okay, we got to make TAA police better. Right. At being like, no, that's a TAA. You better stop.
To understand the new dynamics that have kind of been created now.
Yeah, and so they then messed with the TAA genetics. Yep. Okay. Once they did that, they used assays, which are basically like, you do a bunch of like chemistry and pipetting, and you try to see which proteins are expressed and so on to make sure that this works. The other thing that I found really interesting was, in order to confirm that this worked, they would also infect it with viruses. Okay. This bacteria, and the way the virus works is it hijacks the bacterial machinery to like make more copies of itself. But if we've done this right, then the virus is coming in, looking at this new machinery, like, where's my stop? Like, what's going on?
Where's the factory that?
The factory that I normally do, and then it won't work. Is that kind of cool?
No, that is very intriguing. Yeah, there's so many levels of. Yeah, I mean, we could have multiple podcasts on just individual aspects of the story. Yeah, because there's so many layers of current understanding that are required to even be able to attempt to do this. Yeah,
the hierarchy of processes that happen within a cell at the molecular level. Right. I'm talking like it's just incredible, the understanding that we have. And finally what they did was, so now when they're all done, they have TGA and TAG, which are like, now we can do whatever we want. So then they got these 2, they're called non-standard amino acids, basically made up by us. Right.
And outside of that initial list of 20.
Yeah, and so then they reworked the chemistry that once, there's a part, the ribosome, it sees some 3-letter thing, and then it's like, okay, I'm going to grab this thing. Yep. it started grabbing the right thing, and to grab this new thing that we made up, we have to make it mimic everything else,
right?
So that the ribosome doesn't think anything different. It's just like, oh, this is the one, this is the login key, right? And so they did that, and then finally they created this new E. coli. They call it OCHRE, OCHRE. It's a genomically recoded organism.
Mm-hmm.
It's not the first genomically recoded organism, but this is the first one where they've done all of this, new, like.
Not just changing. No, we're not just doing the changing at the DNA level. It is now producing new proteins that we have made up, and
with new Legos.
With new Legos that we gave it.
Yeah.
And it's doing so on its own. Like, we've created it such that it can self-sustain this new process.
Yeah. And it's like alive.
And it's alive with all of this new internal dynamics, part of which we just made up from whole cloth.
Yeah, it's incredible. It's an incredible story. Like, it's from the Reinhardt and Isaacs lab at Yale University. Well done.
Well, okay.
I mean, yeah.
We're definitely going to almost certainly come back to this story, because I am sure there's going to be a variety
there's going to be. I mean, now that this is the first step in creating a 64 Lego block alphabet.
Right.
Actually, it's got to be 62, because you need one to start, one to stop. Fine. So, but 62 is a lot more than 20.
And if we can, and so like the amount of emergent and derivative properties, behaviors, dynamics that result from tripling the number of Lego blocks that are fundamentally a part of this protein and amino acid generation process. I mean, let the imagination run wild.
Yeah, it's like we've been making houses out of mud. Huts. Like, we've been making mud huts. And all of a sudden now we got steel motherfucker.
Right.
You know, like
you know what this is. I'll end with this note, because
this one thing I wanted to say was, like, its safety concerns are there.
Of course.
Right? But I was reading at the very end of this article, they were talking about, like, how do we do the safety concerns. One of the things that's interesting is you could make these organisms dependent on this new Lego block.
Okay.
Right? And that way, if it gets out, it's just going to die.
Right.
Because these Lego blocks aren't out here.
In the
right? Like, normal E. coli survives everywhere, because it depends on the 20 that are found in us, in plants and everything.
Right.
But, like, if it requires, like, some, like, weird thing that's only found in labs, well, then,
Because I guess the implication of what you're trying to get at is, if this new form of E. coli were to have a leakage problem, we don't necessarily understand what would happen in a free environment, how it would impact flora, fauna, other ecosystems. Let the conspiracy theories run wild. But that's not the third.
One way that we could mandate safety is to say that, like, okay, how about, like, these E. coli, like, in order to even digest sugar or something, like, that protein has one of these.
Yeah, right.
So then it's just going to, like, die.
The world is going to get very, very, very...
I'm sorry. I stopped you in the middle of something.
No, because I always try to think of analogs to help me kind of frame some of these topics. And I was talking to someone the other day that they were talking about... obviously, this is related to the UAP issue. And they kind of made a similar point. It's like, when you look at the periodic table, we have all this certain number of elements that are on there. But we've only really figured out how to engineer those elements at their current, like, at certain isotopic ratios. So it's as an analogy, we sort of have, like, whatever the number is at those set isotopic ratios. But if we could figure out how to create stable states of these other ones, or environments where we can do so,
create the magic number element at, like, 120 or whatever,
then you open up a new set of tools by which you could then engineer things. But we're not using all of the theoretically possible available tools. In that context, there's a similar. It's similar.
It's in that context, I will say that we have a much better understanding of physics and chemistry there.
For sure.
Right? With biology, because there's, like, 10 to the 10 more moving parts. The possibility is, like, it's like we're in biology where physics was, I think, in the 1910s, 1920s.
Okay. Okay.
Yeah.
Which means
Which is like when we were like, oh, it's an electron. What is that?
which means we still have not yet had our, I guess, what is
Manhattan Project.
Yeah, right. Like our
No.
sort of mid 1900s moment where there's just this incredible explosion of like
literally
of like practical applications of the fundamental science.
Yeah, dude, it's
what
nuts.
a great story.
Like, even though this story is kind of old, I think it was February.
Yeah.
But like I came across and I was like,
we have to talk
Lester's
about it.
gotta learn about this.
This is really good.
Crazy.
This is really good. It's unfortunate that it was out of our rivals in New Haven, but
yeah,
we'll they're part of
we'll
part of
give
the
it
club.
to
We'll give it to them.
Yeah, at least it's not Harvard.
Go Tigers. Okay, that I'm gonna have to sleep on that one.